Welcome to I Love Apes. Today we are in Unit 2, Biodiversity, and we're going to look at indicator species, what they are, and how we can use them. So what are they? Well, an indicator species, which is also known as a bioindicator, is basically an organism that we can use to very rapidly check in on the health of an ecosystem. So think of it like a thermometer. You go to the doctor, they take your temperature, and if you have a fever that tells them there's something wrong with you, they don't know exactly what it is, but they know there's something wrong with you. And then they can look further to see how to intervene on your behalf to get you healthy again. It's basically the same idea. So if there is an increase or decrease, depending on the species, that gives us some insight into what's going on with the ecosystem. And hopefully what happens is that it gives us an early enough signal of change that we can intervene and implement some sort of mitigations to prevent greater harm from happening to a particular ecosystem. So in order to be an indicator species, there's just certain qualities that an organism needs to have to make it useful as an indicator species. So let's see what those are. So they have to be particularly sensitive to changes in the environment. That often means you have a narrow range of tolerance. But basically, they are going to respond to changes in the ecosystem before the other things do. So they're more sensitive to these changes, and therefore we'll see change in their populations earlier. We also need them to have low mobility. We don't want them walking out or flying out or slithering out of our ecosystem and back in, because if they do that, maybe what we see is a problem with a different ecosystem, not this one. So we want something that has fairly low mobility. We want it to be something that we can expect to find in the ecosystem. We don't want it to be a particularly rare species, because sometimes those are just hard to find. That kind of defeats the whole purpose. We want something we can find fairly regularly. And we want it to be something it's relatively easy to find and count. So those are the qualities that we need. Fortunately, there's quite a few species that have these characteristics and can then be used. So let's just take a look at some of these species. Well, and there's so many of them. We're just going to look at a few. So let's talk about lichens. So you see this isn't lichen. This is lichen, just saying. You see these things growing on trees and rocks. We talked about them when we discussed primary productivity, but those are mostly encrusting ones. But we have these feathery kind of leafy ones. So what we find is that there's lots of different species of lichens that grow on the trees in a forest, and many of them are particularly sensitive to air quality that could be damaging a forest ecosystem. So we find is that these ones here are particularly sensitive. So these bushy lichens, we'll call them. If you see those, if they are present, what that tells you is, you know what? The air quality is pretty good, especially when it comes to sulfur dioxide, which tends to cause acidity to increase, pH to decrease. If I see these, their presence indicates health. Okay, but if I see a decline in these, and I see there's more of these sort of leafy ones here, then I go, okay, well, if that's the case, then I probably have a growing problem with the air quality. But even these cannot handle, they're not that tolerant of really low pHs, really acidic problems. So there is a problem with the air quality, but it's not a completely catastrophic one at this point. So if I see a transition from this to this, I go, oh, I better intervene and maybe make this coal company put on some sort of mitigation to prevent them from releasing more sulfur dioxide into the forest area. And if I'm down to just these sort of crusty ones, that usually means, if both of these are gone, their absence tells me that I've got a problem with sulfur dioxide or other air toxins in this particular ecosystem. Another really big one that you should definitely know about for this test, and you've probably heard about, is the E. coli bacteria. So E. coli, basically, almost all mammals, including yourself, have them in your gut. We have this symbiotic relationship with them. They just live inside us. They don't cause harm. They're not pathogens, we'll talk about later in the year. They're just in us, okay? They help us digest our food. But the thing is, if you test a water sample and you get this in return, there's no bacteria here, there's no E. coli here, well, that tells you then you probably got a pretty healthy stream system. At least it's healthy in the sense that there's no human or other sewage going into it. It's not being impacted by fecal material from mammals. However, if you see it, that, oh, we got E. coli on this dish from this water we took, it tells you that there's probably going to be somewhere in your ecosystem we're getting discharge into the ecosystem of sewage. So that's another one we can use. Let's keep going. Let's look at another one. Algae is another big one. later in the year, and probably in past years in biology, you talked about this idea of eutrophication. Well, here's how it works. So algae is a group of photosynthetic single-celled organisms that tend to aggregate into these algal mats. They kind of look like a plant, but they're single-celled organisms that group up. Well, anyway, what happens is this. So they're usually not present in very high numbers in a healthy ecosystem. So a healthy aquatic ecosystem tends to have fairly low nutrient levels. There's not a lot of nitrates and phosphates in the water. They're in the soils and the animals and plants instead. So when I look at the rocks growing in a stream bed, in a healthy stream, there's not going to be much algae. So the absence of the indicator species indicates healthy aquatic riverine ecosystem. On the other hand, if I start seeing these algal mats, their presence tells me there's excess nutrients, that maybe farmers are releasing too much fertilizer into the stream, or maybe there's too much soil coming into the stream bringing in nutrients with it. But at any rate, this indicates that we've got a problem, and that problem is nutrients. And again, we see it early enough we can intervene. And another example, we'll make this last one, is apex predators. So apex predators, because they sit at the top of the food web, they are basically bellwethers of everything that happens below them. So if you see a decrease in river otters, brown bears, ospreys, orca, that tells you that there's some kind of problem with the food supply below them. And that could be it could be maybe that there's toxins that are bioaccumulating up in their food web. We'll talk about later in the year. It could just mean that there's a decrease in prey populations. But if I see a decrease in these, I know that there are problems down below, whether it's pollutants or some sort of other environmental harm that's affecting the availability of food for these things. These apex predators are usually pretty easy to spot and monitor. And if you see a decrease in them, that indicates there's an overall problem with the ecosystem. Now, aquatic ecosystems tend to be home, especially riparian ones, river ones, they tend to be home to a number of invertebrates. Many of the things you see flying near a river, in their juvenile stage, they have gills, and they live underwater, and they cling to the bottom of rocks, and they're basically eating zooplankton or algae, okay? So basically, these things, it turns out, are very present in rivers. They're easy to find. They're easy to count. So they certainly make a good indicator species. And many of them have different levels of sensitivity to things like dissolved oxygen or other types of toxins that might be present in our aquatic ecosystem. So I'll give you an example. Tubifex worms. These things are gross. A tubifex worm is extremely tolerant. It can live in environments that have almost no oxygen in them at all. They have very low metabolisms. They can handle very high toxins. So if I see I have a lot of these and not a lot of other things, it tells me I really got a problem, because these things are the last ones to die. So if my river has a lot of these and not a lot of other things, there's a major problem already underway in this ecosystem. On the other hand, if I see stonefly larvae or mayfly larvae, these things are particularly sensitive. They need a lot of oxygen. So if the DO begins to drop, they die off. And by the way, I want to point out, so if I go out to an ecosystem and I sample the water with a dissolved oxygen probe, let's just say, and it looks good to me, I got a great reading here, and I come back a week later, got another great reading, I guess this one is doing great. There's no problem with DO in this river system. However, if I sample these invertebrates and I don't find these things and they're supposed to be there, that tells me sometime between the two samples I took, the DO got low enough to kill these rascals off. So in a way, it's more of using indicator species gives you more of a broader look at the quality of the ecosystem. So you're not just being fooled by taking samples at the wrong time. These things are sort of averaging out over longer periods of time. So let's just look at this. So we can assign them these different riverine invertebrates. We can assign them a tolerance rating. Basically, a low rating means you are not very tolerant. If there's not a lot of oxygen or there is a bit of toxins, you croak. So these ones here, stoneflies, they're the first to go, followed by dobsonfly, caddisfly, crayfish, and I keep going all the way up to tubifex worms, which we assign a number of 10. Now, these are arbitrary numbers, but it's just a way of quantifying the data so that I can assign a number to how healthy the stream is. And we call this the biotic index. So the biotic index is just this method of quantifying the amount of these various organisms I find in a river system and using that to give me a numeric value for the stream's health. So the biotic index is going to be this. I'm going to take the sum of the following. I'm going to take the number of individuals of a particular species, and I'm going to multiply it by their tolerance ratings. And then I'm going to divide, I'm going to sum up these. Let's just say I have 5 of these organisms, so I'm going to have 5 of these qualities added together. And then I'm going to divide by the total number of organisms in my sample. And when I do that, I will get a value here, and that value will allow me to make a determination as to whether the stream is healthy or not. So basically, the higher this value is, because a high tolerance level, so that means that if I have a lot of A's, which is the tolerance rating, high ratings means that you are tolerant, which means you can live in unhealthy systems. So when I get a high biotic index number, it tells me I have worse stream quality. And when I have a low one, it means it's better. So let's see how I go about actually establishing biotic index for a particular stream area. So what I do is I would go out to the stream, and I would use some sort of collection protocol to make sure I'm always going to do it the same way for comparison purposes. And I'm going to collect all the species that I find. And when I do, here's what I came up with. I found 3 species, amphipods, snails, and midgeflies. Just going to keep it easy. I found 4 amphipods. They have a tolerance rating of 5. I found 7 snails, tolerance rating 6, and 3 midgeflies, which have a tolerance rating of 7. So now I'm just going to use this in that biotic index equation to see what value I come up with. So the biotic index is going to be the sum of the number of individuals, which would be, in this case, 4 amphipods, times the tolerance rating of that particular species, 5. And I'm just going to sum it up for each of these. So I'd say I have, for amphipod, I have, the number was 4, the tolerance rating was 5. And then I'm going to add that to the other one. I have 7 snails, tolerance rating 6. Plus, I have 3 midgeflies, tolerance rating 7. The total number of organisms that I collected was 14. So I'm going to put 14 down below here. That's how you do it. And it basically gives us a weighted average. And when I do this, I find my biotic index is 5.93. So once I have established what that biotic index is, I can use it to compare it to other streams. So basically, you could say if I have a biotic index that's less than 3.75, I have excellent water quality. I have good DO. I don't have much organic pollutants. And what I find is, as these numbers get bigger, it's becoming worse. So I think what I had before was in this range here. So that stream was, it's fair. That means there's definitely some pollution likely to be in that stream. Otherwise, we would have found more of these lower tolerance species. But it's not like super poor like here. Okay, so that's basically how we're going to use it. Thanks for listening. That's indicator species and how we use them. Hey, thanks for watching. If you like this video, please go ahead and like this video. If there's something I do to make this better, or if there's another video you'd like me to make, please drop me a comment. You should probably check the description below because I do sometimes post problems that could help you do better on your AP test. And of course, you know it's coming. Please hit that subscribe button for me.